Okay, welcome back. Are we ready to start translating? Translation is going to start at the N terminus of the polypeptide and add amino acids until it reaches the C terminus. On mRNA, we're going in the 5' to 3' direction. Translation is going to be divided into 3 stages. One is initiation, then elongation, then termination. In initiation, the mRNA is going to bind to the ribosome, and then we're going to get it positioned all the right way on that ribosome. This is really what we're talking about in this video. Then, after initiation happens, elongation happens when you're bringing in amino acids and joining them one at a time with peptide bonds. And then the last step is termination. Once you have made your polypeptide, it's a stop codon comes in and releases the entire molecules from each other. Initiation of translation in bacteria and in eukaryotes is different. In bacteria, we can divide initiation into 3 steps. In it, the initiation factors IF1, IF2, and IF3 bind to the small subunit of the ribosome, and GTP will be bound to IF2. So we have our small subunit, we have our GTP. The mRNA and the tRNA with the start codon, with the first amino acid, are going to come in and bind to the 30S subunit. The mRNA is going to come to the proper sequence because of this area called the Shine-Dalgarno sequence. The Shine-Dalgarno sequence is going to be 3 to 9 purines upstream from the start. These purines are going to base pair with the 3' end of the 16S rRNA, which allows the mRNA and the ribosome to be formed together. So this is what the mRNA binding site is here. When mRNA is bound to the subunit, to the small subunit, it's going to be arranged so that the start codon is here at the P site. The start tRNA carrying N-formyl methionine, or FMET, is going to come in to initiate translation. And it only has a carboxyl group where it can bind to another amino acid. So it can only bind in that direction. So tRNA with N-formyl methionine is the initiator tRNA. That is the tRNA that we use to get the whole process started. The initiator tRNA is going to bind to the P site of the small subunit, and it does this by the action of IF2. When you have the small ribosomal subunit, the initiation factors, mRNA, and tRNA, this is the 30S initiation complex. Then IF3 is going to be released, and the 30S initiation complex is able to bind to the large subunit. And now you have the 70S initiation complex. This large 50S subunit is going to hydrolyze IF2 and GTP, which is going to cause IF1 and IF2 to leave. So the initiation factors that you needed to start it are able to leave. They're released once you've got this whole ribosome set up. So in eukaryotes and archaea, we're using the start codon methionine, which is a typical amino acid, rather than this atypical N-formyl methionine. And the initiation factors that we use are called EIFs, or eukaryotic initiation factors. And there's more than 3 of these. There's about a dozen of these. We have different genes for the initiator tRNA methionine and in initiation, and the tRNA methionine used in elongation. And the initiator tRNA methionine is going to favor binding to the initiation factor EIF2 and prevent binding to the elongation machinery. The EIF2 will be attached to GTP, and it's able to bind to the tRNA methionine before the tRNA has bound. And the tRNA will bind to the small ribosomal subunit with the other initiation factors, so EIF1, EIF3, and EIF5, and then it'll bind to EIF1A, and this creates a 43S pre-initiation complex. mRNA is going to bind to the 43S pre-initiation complex. So I've got mRNA added to it. The mRNA 5' cap will be recognized by one of the initiation factors, EIF4E. That's going to bring in EIF4G, which makes translation efficient. And then EIF4A and EIF4B come in. And EIF4A is going to be a helicase, which helps separate those nucleotides. Then those two complexes are joining because there's the EIF4G and EIF3 will interact with this small subunit and the mRNA. EIF2 will hydrolyze associated GTP. A lot of the initiation factors can leave. And then the EIF5B with GTP will bind. After binding to mRNA, the small ribosomal subunit, which has tRNA methionine, will scan along the mRNA, and when it finds that first AUG, it's going to start translation. On either side of the start codon, there are some nucleotides that are involved in the start site recognition. This one sequence, the Kozak sequence, is going to be ACC AUGG. And within this, you can see the AUG, which is your start codon. Once that initiator tRNA is paired with the start codon, the large ribosomal subunit joins a complex, similar to how we saw happen in proteins and prokaryotes. And EIF5B will facilitate this reaction. It'll hydrolyze its GTP along the other initiation factors to dissociate. Now you have a fully formed ribosome. In eukaryotic initiation, the mRNA is going to be polyadenylated or have a lot of adenines at its 3' end. There's a poly-A binding protein, or PABP, that will bind to that poly-A stretch, and it's also able to bind to an initiation factor, EIF4G. And that EIF4G is one of the initiation factors involved in initiating translation. So the 3' end of mRNA helps stabilize the 5' end and make it ready for initiation to occur. Okay, we've made our ribosomes. We've got everything in process, in place. In our next video, we're going to talk about actually getting that protein made. I'll see you then.